Hello, my name is Tony McLean. I'm from Sydney, Australia. And the presentation that I'm giving today is what you should know about genomics, and it's directed to critical care physicians. And of course, the first question is how can we fit that into a short session that we have available today? And I'm going to concentrate on 6 points really. One is why we should learn about it. What are some important terms? Not them all, but just a few. Let's talk about inherited disorders, transcriptonomics, epigenetics, and other. So the first point really is, why should we actually learn anything about genomics? At the end of the day, the intensive care and emergency medicine curriculum has so many components now. Why would we add something which, is perceived to be quite complicated at times, such as genomics? And the reality is shown here. This is a slide of a patient in our ICU with COVID, and I know that many of you around the world have had extraordinary exposure to these patients. Fortunately, here in Sydney to date, we've only had a small number, but if you had a patient in the unit, then in fact we use genetics in a number of ways. First of all, the pathogen PCR to diagnose it. We now do a lot of work on the immune response of the host to such an infection. As we will see, there's an hereditary susceptibility in some ways for people who actually have severe COVID. Maybe the person is on immune modulating therapy, such as having an underlying lymphoma or cancer. We now know that the world of comorbidities are very important, while some people are more susceptible to infection than the others, and then epigenetics comes into play. Transcriptonomics, in fact, is an area that has been of immense interest to critical care physician researchers over the past few years, and an unknown quantity, but one waiting to be discovered in much greater detail, is pharmacogenetics. So let's start with some important terms. We don't have to know many terms, but terms like what's the difference between genomics and genetics is the most common question. And the easiest answer is that genetics is the study of hereditary, and as shown down here, you're really dealing with a single gene, and that single gene can determine what your genetic makeup is and how you respond to infections and the like. Whereas genomics addresses a number of genes, in fact, all the genes, and their interrelationships in order to identify their combined influence on the growth and development of the organism. And of course, genomics has only been available since we have techniques that can deal with this enormous amount of information. Molecular biology is a term often used, and molecular biology is the field of biology that uses all these areas, such as molecular biology, genetics, proteins, function, and so that's why you hear the term used as well. Phenotype, an observed trait. Genotype, the genetic information defining the phenotype. Alleles, alternative forms of a gene or a genetic marker, and a haplotype, which is a group of genes within an organism that's inherited together from a single parent. Polymorphism is where you have a genetic variant which, although it's very rare, has a frequency of greater than 1%, independent on whether it has any influence on the organism or not. A single nucleotide polymorphism is called a SNP, and it's where the alleles vary just by one single nucleotide base. A mutation is where there's an occurrence of change in the genomic sequence or a resultant change itself. And just going back to allele, of course, if we have a variant of a gene, the variants can be in a number of ways. Either DNA sequence, whether you're looking about different haplotypes, functional versus wild type, parental origin, whether it comes from your mother or your father. So the allele can be, in fact, a descriptive term in a number of different ways. So let's get on to inherited disorders. And I'm just going to touch upon this briefly. And it's really important because this is a very interesting study back in 1988, which showed that if your parents, these are children who are adopted out, they did not live with their parents, they live with adopted parents. But if your biological parents died from infection, you had a nearly 6-fold increase of you yourself dying from infection. So there's something in the genes that makes us more susceptible which in fact is not the same as with adoptive parents. So if we look at hereditary, we need to look at the DNA. And the DNA, of course, is placed within the, as shown here in the picture, is shown in a nucleus. The DNA splits into two, and then it's translated, it's transcripted to RNA. The RNA leaves the nucleus and goes into the cytoplasm, and there in the cytoplasm, protein is produced. And then there's some post-translational modifications where you have modified protein. And of course, inherited problems we have come from changes in the DNA. And I'm just giving 3 examples that would affect intensive care patients. One is an out-of-hospital cardiac arrest. The Brugada syndrome is shown with these classical changes with the partial right bundle branch block pattern in V1 to V3. You have this elevation of the R wave coming in a sloping fashion towards the T wave, and it's autosomal dominant inheritance. It's a calcium channel abnormality. It occurs at any age, but typically sudden death occurs around the age of 40. A second one we come across are bleeding disorders, and I just use one example here, which is the factor V Leiden mutation. And this is where the F5 gene, which normally causes thrombosis, which is then controlled by activated protein C, the factor V Leiden mutation interferes with activated protein C in activating the clots, so the clots become very large. And overall in the population, your chance of developing a DVT is 1 in 1,000 people per year. If you're heterozygous, it's 3 to 8. But if you're homozygous, in fact, it comes up to about 80 DVT chance out of 1,000 people per year. And for those of you who are traveling through the Czech Republic, in the city of Brno is a garden here which Gregor Mendel used to use in developing the genetic therapy, a genetic theory, in fact, on the heterozygous and homozygous features. A third one is susceptibility to sepsis. And what I put down here is a paper that was just published last week in JAMA to show that 4 young adult patients who developed severe COVID-19 infection in Holland, they were found to have toll-like receptor 7 abnormalities on the X chromosome, and this abnormality resulted in these people developing severe infection because of the interference with the innate immune system. The fourth area is one, in fact, is a little more difficult to come to grips with, but the modern intensivist and emergency physician really has to understand this because it really is changing what's happening in our world, and that's transcriptonomics. And what we're talking about with transcriptonomics is actually the RNA. Remember, the DNA is inherited in differences. This is what we use. We measure the RNA to identify how the patient is coping. And in fact, just to remind ourselves, the mRNA comes from the nucleus into the cytoplasm, and what we do is we measure this mRNA. And the study of transcriptonomics, as shown in this Nature definition, is in fact, it's all the genes that are expressed by a particular influence on the cell. And only by using high throughput methods such as microarray or next generation sequencing can we actually identify what these expressions are. Here's one from our lab, for example, which was published in the European Respiratory Journal in 2017. These are from the days in H1N1 influenza. We looked at these patients, for example. We looked at all the genes that were upregulated, which is shown by the red, and the genes which were downregulated, shown by the green. And you can see the difference between community-acquired pneumonia with bacteria and those with H1N1. On day 1, they have a very different genetic signature, with all these genes being upregulated in the community-acquired pneumonia, but being downregulated in H1N1. And the reverse occurred with all these other genes, which were upregulated with influenza and downregulated in community-acquired pneumonia. And by 5 days, this distinct genetic signature was starting to blur. But in the first few days using this technique, if you had a patient with whiteout of the lung on a ventilator, you weren't too sure of the diagnosis, we can tell you whether it was H1N1 or bacterial infection. There are lots of studies on this. This is a very important one, a landmark one, published in the Lancet Respiratory Medicine by the Mars Consortium, looking at genomic endotypes in sepsis. And what they found that in using transcriptonomics, you could divide them into Mars 1, 2, 3, and 4, and they had a different mortality associated with a particular genetic expression. Another one is with Timothy Sweeney's group, looking at transcriptonomics across a whole number of multiple databases, such as these ones here, with 8 countries for the initial investigation, and then 600 patients for validation. And they identified 3 subsets, which they called coagulopathic with high mortality, adaptive with low mortality, and inflammopathic. The trouble here, it's all big data, and you need to really be able to analyze it well. So we can have a single gene which does it. Moving on, epigenetics. Epigenetics is in fact where you have things which influence the DNA, which is outside the DNA structure. We know now there's a lot of microRNAs which can affect the coding of the genome, and we also know that epigenetics involves changes of the structure around the DNA helix. DNA is coiled around the histones, creating structures called nucleosomes, and we know that epigenetic changes come by changes in the histones, the methylation of the DNA, and RNA activities. Where does this affect us? It's because this is where the comorbidities come in. And if you have comorbidities such as cancer, cardiovascular disease, diabetes, autoimmune disease, and sepsis, you actually affect the expression of the DNA and therefore the response of the host cell. So genomics, there's a quick rundown. It's not complete. There's certainly a number of other things which I haven't touched upon. I'm not saying they're not important, but they're less well developed. But over the coming years, you will find that this is an important part of the treatment of a patient in ICU. One is pharmacogenetics. We now know that a lot of the drugs that we use have influences, particularly if they are used together. We haven't talked about pathogen genetics, which is the diagnosis of pathogens on a regular basis, just not with a swab in the nasal passages looking for COVID or the like, but in a number of other ways. And mitochondrial genes is another area as well. So genetics, in fact, and genomics is becoming an increasing part of what we do in intensive care and emergency medicine. And a basic understanding is really important to moving into the future in dealing with critical care illness. And on that, like that, I'd like to thank you for listening, and I'd like to finish on that note, just saying the reasons why we have to know it, what the important terms are, the inherited disorders, transcriptonomics, epigenetics, and of course the host of other things which we'll become familiar with over the coming years. And I just complete with my finishing slide, which is just up the road here where I live in the Blue Mountains, and this is Three Sisters in the Blue Mountains, just west of Sydney. Thank you.